Hello everyone and welcome to today's session. And today we'll be discussing the theory related to the chapter evolution. In this session we'll be discussing NCERT section 6.6 and 6.1, and this is the part 4 of the lecture. This is the disclaimer. So let's understand what are the objectives of today's session. So first we will compare the Darwin's theory of natural selection with the Hugo de Vries mutation theory of sudden changes. In this we'll understand the concept of saltation given by Hugo de Vries. Okay, we will also understand how mutation and genetic recombination and natural selection work together in the evolution. Okay, we'll also talk about the Hardy-Weinberg principle. Okay, we'll talk about the equation p square plus 2p square plus q square. Okay, we'll also understand how to calculate the genetic equilibrium in the populations. Then we'll also talk about the 5 factors which disrupt the Hardy-Weinberg equilibrium. In the end, we'll be also talking about the types of the natural selection. Okay, so today we'll be talking about mechanism of evolution, which is NCERT section 6.6. Then we'll talk about the Hardy-Weinberg principle, which is NCERT section 6.7. Then we'll summarize this entire session. We'll also discuss some NCERT-based questions related to these topics, and then we'll discuss some keynotes in which there will be some interesting information as well. These keynotes will help you memorize these topics as well. Okay, in the end, there will be some interesting questions that will enhance your critical thinking ability. Okay, we'll be discussing about the mechanism of evolution today. So first, let's talk about the Hugo de Vries idea. So he was working on evening primrose. What is the scientific name of primrose? It is Enothera lamarckiana. Okay. Now, Hugo de Vries proposed the mutation theory of evolution. Now let's understand what is mutation theory of evolution. So his key idea was that evolution is caused by mutations. So we all know mutation can cause variations. Okay. So according to Hugo de Vries, evolution is not gradual. So Darwin said that evolution takes millions of years. It is gradual. These variations, they accumulate over generations, leading to the formation of a new species. But according to Hugo de Vries, evolution is caused due to mutations. Okay, what kind of mutations? So according to him, mutation is large. They are sudden, they are random and directionless changes in the population. Okay. So these mutations are large. You can see they are random, they are directionless, and they occur suddenly. Clear? Now, he gave this, you can say, concept of this large and random mutations as saltation. So what do you mean by saltation? So Hugo de Vries believed that mutation caused speciation, means formation of a new species, and hence called as saltation. What is saltation? Single step large mutation. Means the evolution is occurring in just a single step. A new species is forming in just a single step due to one large mutation. Okay, and these mutations are sudden, these are random, these are directionless. Okay. Now what was Darwin's idea? So his key idea was that variations as the mechanism of evolution. So what did Darwin said? So Darwin said that variations, they keep on accumulating over generations, and this leads to the formation of a new species. So he said variations are small and directional, and evolution is a gradual process. It is a slow process, and these variations which provide survival advantage, which help the organism or a species to survive in the changed environment, these variations are selected over the generations. These variations accumulate and lead to the formation of new species. Okay, so according to Darwin, evolution is gradual with small heritable variations. Okay, these variations are, you can say, passed on to the next generation, and they keep on accumulating over generations. Let's talk about the modern synthesis. Now, while mutation is the ultimate source of all genetic variation, okay, so by mutation, what happens? There is a change in the DNA sequence. Okay, now this new sequence can code for some another product. So mutation, we are saying it is an ultimate source of genetic variation. But apart from mutation, recombination can also lead to variation. Okay, so variation can be due to mutation and as well as genetic recombination as well. Okay, so genetic recombination occurs during the sexual reproduction. So we are saying here that the evolution is now understood to be a combination of mutation as well as genetic recombination and natural selection acting on this variation. So nature is selecting the variations okay, which are beneficial, which give survival advantage. These variations selected in the next generation, they are, you can say, inherited in the next generation. So this principle states that, okay, now we are talking about the Hardy-Weinberg principle. Okay, what does Hardy principle state? Okay, so this principle states that the allele frequencies in a population are stable and is constant from generation to generation. Okay, so we can say the allele frequencies or the gene frequencies, they remain stable from one generation to next generation. There is no change in the allele frequencies. Okay, so this principle is very important, very crucial for understanding how populations evolve over time. Okay, now the gene pool, what is gene pool? It is the total genes and their alleles in a population. So usually a single gene, okay, one gene have two alleles. Okay, it can have more than two alleles also, but usually a single gene have two alleles. So what is gene pool? It is the total genes and their alleles in a population. This gene pool remains constant. Okay, this is known as genetic equilibrium, when the gene pool remains constant. Clear? So when this equilibrium is disturbed, then we can say that population is evolving. Now let's talk about the mathematical representation of the Hardy-Weinberg principle. So we are talking about a diploid organism. A diploid organism usually have two alleles. Clear? A single gene have two alleles. These alleles, they are present at a particular locus on the chromosome. Locus is a position. Okay, suppose there is a gene which have two alleles, A and small a. Now let the frequency of allele capital A be P and let the frequency of allele small a be Q. Now the frequencies of the genotype capital A, small a and small a are. Okay, the frequency of capital A will be P square. Then the frequency of capital A, small a or small a, capital A will be 2PQ. Okay, that's where we're writing 2PQ. There can be two combination, A and small a and A. And then last frequency of small a is Q square. Clear? Now sum of all the genotype frequencies equals to 1. Means P square plus 2P square, P square plus 2PQ plus Q square is equal to 1, which is basically P plus Q whole square. Okay, so this equation P square plus 2PQ plus Q square, this is the binomial expansion of P plus Q whole square. Okay, and we say that the sum of all the genotypic frequencies will be equal to 1. Now, what is the significance of Hardy-Weinberg equilibrium or Hardy-Weinberg principle? So, we can say that if the population is in Hardy-Weinberg equilibrium, it is not evolving. Okay, there is no change happening in the population. If this equilibrium is disturbed, then we say that the population is evolving. So, disturbance in the genetic equilibrium or Hardy-Weinberg equilibrium indicates evolution. Okay. We can also say that if the measured frequency of genotypes, means if the measured genotypic frequency in a population deviate from the expected genotypic frequencies, it indicates the extent of the evolution. Okay, or you can say the difference between the measured genotypic frequency and the expected genotypic frequencies, this difference indicate the extent of the evolution. And if there is a deviation, it means that the population is evolving. Now let's talk about the factors affecting the Hardy-Weinberg equilibrium. Okay, what are these factors? So basically there are 5 factors which affects the Hardy-Weinberg equilibrium. Okay, what are these 5 factors? First is gene migration or the gene flow. Okay, let's talk about this. Now, population movement can alter the gene frequencies. Okay, so they can be immigration or they can be, people can leave the population or they enter into the population. They can be migration or immigration. Clear? So this can change the gene frequencies because genes are added in the population when the people are entering a certain population, or the genes can be, you can say, lost when the people are moving out of the population. So this can alter the gene frequencies. So there is change in both the source and the new population. Okay, the gene frequencies of the source population, new population, both in both of them the gene frequency can change or the allele frequency can change. Now the new gene or alleles are introduced to a new population or they are lost from the original population. Gene frequencies are being changed. Now repeated gene migration leads to gene flow. Okay, clear? If the migration is happening again and again, so it will lead to gene flow and there will be a change in the gene frequencies. So this can lead to change in the genetic equilibrium and the population can evolve. Second is genetic drift. So genetic drift is basically change in the allele frequencies by chance alone. And this is especially significant in the case of small populations, not in larger populations. It is significant in smaller populations. Okay. Now let's here talk about the founder effect. Okay, what is founder effect? Now, genetic drift changes the allele frequencies in the small population. So genetic drift basically affects the small populations. Okay. These isolated small populations show founder effect with limited genetic diversity. So the population is already small, means they have limited genetic diversity. Okay, so these small populations, they show founder effect. Okay, now genetic drift combined with selection and isolation can lead to population divergence. So there is genetic drift happening. This is combined with the natural selection as well as isolation of that species, of that population, and this can lead to population divergence, means there was original source population. Okay, there was original population and then there was newly formed population. This is known as population divergence. Let's understand founder effect with the help of example. When a small group of individuals, which are known as the founders, they breaks off from the larger population. Okay, So they are moving out of the original population and they are starting a new one. The new population's gene pool may be different from the source of population. Okay, so the allele frequencies will be different in the new population as compared to the original population. Okay, so you can see in this image there is a parent population, also known as the original population or source population. Okay, now the genetic diversity is shown here by these various colored circles, okay, within this parent population. Now you can see here a certain group of individuals, they are migrating to another area. This is migrating population. Now they have colonized a certain area. Okay, now you can see they will increase in number. So you can see this is the population after a couple of generations, and then you can see these founder populations after a few more generations. Now you can see this new population. This has different allele frequencies as compared to the parent population, and this is just by chance. Okay, so just by chance there can be a change in the allele frequencies, as in the case of founder effect. Okay, so these small group of individuals which initially migrated, these are known as the founders. Founders will form the founder population. Okay, so this can also lead to change in the allele frequencies. Okay, now let's move on. Next factor which affects Hardy-Weinberg equilibrium is mutation. Clear? So it is the ultimate source of new alleles. So see, by mutation what happens? There is a change in the DNA sequence. There is a change in the nucleotide sequence. Okay, so mutation can give rise to new traits also. It can give rise to new alleles also. Okay, basically alleles, they are the you can say different forms of a same gene. These alleles are different from each other. Okay, what is the difference? Difference in the nucleotide sequence. And this difference in nucleotide sequence can arise due to mutation. The mutation leads to variation. It is a source of new alleles. So because of this, the allele frequency can change. Clear? Because this new allele can provide survival advantage, and this can be selected to the next generation. Next is genetic recombination. This is also a source of variation. In recombination, what happens? There is the exchange of the genetic material. Clear? Now you can see shuffling of existing alleles during the meiosis. In meiosis, what happens? Genetic recombination occurs, crossing over occurs. And due to this, there is a shuffling of the existing alleles. So new combinations are made, which leads to variation. Okay, so this leads to introduction of new alleles, altering the allele frequencies. Okay. Now, next factor which influences the Hardy-Weinberg equilibrium is the natural selection. Okay. So, natural selection is nature is selecting. Okay. Now, these variations which provide survival advantage, these variations are selected in the next generation. Okay, so heritable variations that improve survival are favored. So nature will select only that characteristics, that variations which provide survival advantage to the organism or species. They are passed on to the next generation. Okay, so this will lead to increased reproduction and more offerings of the species which is being selected. Okay, because this species can now, or this species has that variation which help it to survive in that environment. So this organism or this species will produce more offerings. It will be more fitter as compared to the other species. Okay. Now, differential survival and reproduction based on heritable variation. Okay, so there is differential survival, means the organism or species which have certain variations or which have certain traits which help them to survive in the environment, that will survive, and rest of the species will perish, or the organism will perish. And the organism which is being selected, that will produce more offerings, their population will increase, their number will increase, and rest of the organism will perish. Now, natural selection linked to better reproduction leads to different population. Okay, so natural selection can also lead to change in the allele frequencies, because nature is selecting that alleles. Okay, nature is selecting those alleles or genes or variations which provide survival advantage. These genes or alleles then become fixed in the next generation because they are being keep on selecting by the nature. So ultimately they become fixed in the population, and the allele frequency will change in the new population. Okay, because this new population will be formed by that organisms which were selected by the nature. Okay, now let's talk about types of natural selection. There are three types of natural selection. First is stabilizing selection, second is directional selection, and third one is disruptive selection. Let's talk about all of these three types of selection. Okay, so first is stabilizing selection. Okay, so what is stabilizing selection? So in stabilizing selection, the average or intermediate phenotype is being selected and the extreme variants are eliminated. Okay, and the peak of the distribution curve gets higher and narrower because the intermediate phenotype is being selected and extreme phenotypes or variants are eliminated. Example, in humans, the average birth weight babies have a higher survival rate than very small or very large babies. This is an example of stabilizing selection. So in this image, you can see the stabilizing selection here. Okay. So initially, you have this original population. Okay. Then you can see in image A, the peaks are getting higher and narrower. This is in the case of stabilizing selection because intermediate phenotype is being selected. Here stabilizing selection is being acted upon on the original population. Next one is directional selection. So in directional selection, one extreme phenotype is favored over the others, and this causes the allele frequency to shift over time in one direction. Okay, now the peak of the distribution curve shifts in one direction. Here, example is industrial melanism in peppered moth, where the dark phenotype was selected after the industrialization. Here you can see in image B, this is the image showing you directional selection. Okay, here one of the phenotype is being selected. Okay, so only one of the allele, you can say, is being selected. So this causes the allele frequencies to shift over time in one direction. Okay, because one phenotype is being favored. Okay, so the original distribution curve, this is moving in the one direction. Here, now let's talk about the third type of selection here. Third is disruptive selection. Now here both the extreme phenotypes are being favored over the intermediate one. The population may split into two distinct groups. Okay, that means the single peak splits into two. Example is in a population of birds, if both were small and very large. Okay, seeds are available, but medium seeds are not. Birds with very small or very large beaks would be favored. So, in an area, there were two kinds of seed present: very small seeds and very large seeds. Medium seeds were not present. So, only that birds were selected or they survived which had very small beaks or very large beaks. Okay, medium sized beak, they didn't survive. They were not selected because medium sized seeds were not available in that area. So you can see in this image C, it is showing you disruptive selection. Here the extreme phenotypes are being selected over the intermediate, and because of this, initial single peak is divided into two peaks. Clear? Now we have discussed the theory. Let's summarize this entire lecture. We talked about the mechanism of evolution. So Hugo de Vries, he gave the mutation theory. Okay, so he said that these mutations are sudden, they are large, and the evolution of a new species occurs in single step. So he called it saltation. Single step large mutation leads to evolution of a species. Okay. Now Darwin said that the evolution is gradual. These variations, they are very small, they are directional. These variations, they accumulate over generations and lead to the formation of a new species. Now what do modern synthesis say? So it says that evolution is due to combination of mutation, genetic recombination and the natural selection as well, which is acting on these variations. So mutations and recombination, they cause variations, and these variations are selected by the nature. Okay, which is natural selection. We also talked about the Hardy-Weinberg principle. So this principle says that allele frequencies remain stable in the population over the generations. Okay, if there is a change in this equilibrium, then we can say the population is evolving. Mathematical representation of Hardy-Weinberg principle is p square plus 2b square plus 2pq plus q square is equal to 1. Now population in equilibrium is not evolving. Okay, if there is a change in this equilibrium, this means population is evolving. We also talked about the factors disturbing the Hardy-Weinberg equilibrium. First is gene migration or gene flow, means movement between the populations. Second one is genetic drift, which means random changes in the small populations. Okay, within this we'll also talk about the founder's effect. Third one is the mutation, which is the source of new alleles. And then genetic recombination, which causes shuffling of the existing alleles, and due to recombination new, you can say new combinations are formed. The last one is the natural selection, which leads to differential survival based on traits. Okay, so only that traits are being selected by the nature which gives survival advantage. These traits are inherited in the next generation. These traits are controlled by genes. So you can say genes which provide survival advantage, they are passed on to the next generation. And so on, the certain gene is being fixed in the population. So the allele frequency or the gene frequency changes. Okay, this leads to evolution of a new species. We also talked about the type of natural selection. First is stabilizing selection. It favors average phenotype, eliminates the extreme phenotypes or extreme variants. Second is directional selection, favors one extreme phenotype. Okay, so here the population shifts in one direction. The distribution curve shifts in one direction towards one of the phenotype. Third one is disruptive selection. It favors both the extreme phenotypes. Okay, creates two distinct groups. We also talk about the founder effect. So it is a special case of genetic drift only. Clear? Now, small group starts a new population. So these founders, they start a new population. So these initially a group of individuals, they migrate to a new area. These are known as founders. Okay, so here you can say the allele frequency of the new population changes by chance. Okay, so this can lead to formation of new species as well, because allele frequencies in the new population can change by chance. Okay, this occurs in a small population. Let's discuss some NCERT based questions. So first question is, what is the key difference between Hugo de Vries and Darwin's view on mechanism of evolution? Okay, what will be the answer here? Let's discuss the answer. Okay, so Darwin's theory emphasized slow, gradual changes and the survival of the fittest. Whereas de Vries proposed that new species arise from sudden and significant mutations. Okay, so Darwin said that the evolution is gradual. Okay, it is a very slow process. Variations accumulate over time. Okay, these variations selected by the nature. Clear? In contrast, Hugo de Vries said that speciation can occur in one single step due to one large mutation. Okay, he said mutations are random, they are large, they are directionless. So this is the table showing you differences between the theories given by Charles Darwin and Hugo de Vries. So according to Charles Darwin, mechanism of evolution is natural selection. According to Hugo de Vries, mechanism of evolution is mutation. Now, source of variation in Charles Darwin's theory was gradual accumulation of small inherited changes. Okay, Hugo de Vries said there are sudden and large mutations and variations are discontinuous. Clear? Discontinuous variations. Now it means new traits can arise suddenly. Now, what is the process of change in case of Charles Darwin? So it is slow and continuous. So evolution is very slow. It is a gradual process. Okay, these changes occurs very slowly, but it is continuous. In the case of Hugo de Vries, these changes are very rapid. Okay, and occasional. Then Charles Darwin, what was the focus of study? Adaptive traits and survival advantage. And Hugo de Vries, in case of Hugo de Vries, focus of study was the role of new trait arising through mutations. Okay, so Hugo de Vries said mutation can give rise to new phenotypes. Okay, now what are the example organisms? Okay, which show natural selection? Darwin finches, which show adaptive radiation. Okay, various species examples as well, which exist in the nature. Now, Hugo de Vries, he worked on the evening primrose, whose scientific name is Enothera. Next question, what is Hardy-Weinberg equilibrium and what does p square plus q 2pq plus q square is equal to 1 represent? So let's have a look at the answer. Okay, so Hardy-Weinberg equilibrium or this principle says that allele frequencies in a population are stable and it is constant from generation to generation. Now the gene pool, which is total genes and their alleles in a population, remains constant. This is known as genetic equilibrium and sum total of all the allele frequencies will be equal to 1. Clear? Now the formula which represents genotypic frequencies are given below. Okay, so p square is equal to frequency of homozygous dominant, which is capital A, capital A. Then 2pq is the frequency of heterozygous, which is capital A, small a. Then q square is the frequency of homozygous recessive, which is small a, small a. The total of all the frequencies will be equal to 1. Okay. The next question, distinguish between stabilizing and disruptive selection with an example. Okay, so let's have a look at the answer. So what is the difference? So stabilizing selection, how can you define it? Stabilizing selection favors intermediate traits, reducing variation. And disruptive selection, what happens here? Extreme traits are being favored, and this increases the variation. Okay. In stabilizing selection, what is the effect on population? Narrows the phenotypic range around the optimal trait. In disruptive selection, it split the population into two groups with distinct extremes. Okay, in stabilizing selection, what is the result on variation? Decreases the genetic diversity because intermediate is being favored, and in disruptive selection, it increases the genetic diversity because extreme phenotypes are being favored. Okay, now let's talk about the examples of stabilizing selection. It is human birth weight, very low or high, or very high weights have lower survival, intermediate weights are favored. Then, if we talk about the disruptive selection, examples are Darwin finches. Birds with either very large or very small beaks survive better than those with the medium beaks. Clear? Now, basic evolutionary mechanism. Okay, let's discuss some keynotes now. So, we talked about the mechanism of evolution today. Okay, so what was the mechanism of evolution given by Hugo de Vries? So he said he gave mutation theory based on Evening Primrose. Okay, he believed in certain large changes, which is known as saltation. Okay, so he said the new species can evolve due to a single large mutation. Okay, it can evolve in just single step due to one large mutation. So here you can see for your NEET exam, definition of saltation is necessary, and the contrast of Hugo de Vries theory with Darwin's theory is also very important. Then if we talk about Darwin mechanism of evolution, so he said natural selection is the mechanism of evolution. Okay, he said evolution is through small and gradual variations which accumulate over generations. And these are directional changes, variations which give survival advantage. These are selected into the next generation. What is important for NEET exam? Comparison between Darwin and Hugo de Vries theory. Then we'll also talk about the modern synthesis. So he said the evolution is due to a combination of mutation, genetic recombination, and then natural selection act on the variations which arise due to genetic recombinations and mutations. Here, understanding of combined evolutionary forces is very important for the NEET exam. Then we also talk about the Hardy-Weinberg principle. So it says that the allele frequency remains stable over generations. You should know the mathematical expression of the Hardy-Weinberg principle, which is p square plus 2p plus 2q square is equal to 1. And the Hardy-Weinberg principle basically represents the genetic equilibrium, that the gene pool will remain constant. Okay, so here you should know about the Hardy-Weinberg equation and the conditions required to maintain the genetic equilibrium. Okay, we also talk about population changes and selection. Okay, these are the factors which affects the Hardy-Weinberg equilibrium. First is gene migration or gene flow. What is gene migration? Movement of genes between the population. It alters the gene frequencies. For the NEET exam, you should know the definition and effects on the population. We also, second factor was the genetic drift, which is random change in the allele frequencies. Within this, you should know about the founder effect. Here, the small population is establishing a new colony. Okay, you should know about the founder effects, examples, and impact on the small populations. Then the next factor was mutation. Okay, mutation is a source of new alleles, and recombination was another factor which caused shuffling of the existing alleles. Then we also talk about the natural selection, which is the fifth factor. We talked about the types of the natural selection. First, we talked about the stabilizing natural selection, which favors the average trait. Then is the directional selection, which favors the extreme trait. Okay, it favors one of the phenotype actually. So here the curve, it moves in one direction, and disruptive, it favors both the extreme phenotypes. Okay, so you should know about all of these type of selection with examples. You should know the distribution curves, and interstitial mechanism example is very important. Okay, so now there are some interesting questions for you. First is, how do bottleneck events differ from founder effects in their impact on the allele frequency? So you should know the difference between bottleneck events and the founder effect. Next one, why is random mating a crucial assumption in Hardy-Weinberg population? Okay, You should think about these questions. Next one, can genetic drift have a stronger effect on evolution than natural selection? And in what circumstances? Next one, how does inbreeding affect Hardy-Weinberg equilibrium frequencies? Okay. Next one, why do larger population tend to maintain genetic variation better than the smaller ones? Yes, so with this we have completed the theory. Now let's discuss some DPP related to these topics. So let's discuss the first question. Which of the following statement correctly differentiate Darwinian variations from mutations according to Hugo de Vries? Okay, so we have four statements here. First one is Darwinian variations are large and sudden, while mutations are small and directional. Is this true? Next one, Darwinian variations are small and directional, while mutations are random and directionless. Next one, Darwinian variations are random and directionless, while mutations are small and directional. And the fourth one is Darwinian variations are caused by environmental factors, while mutations are genetic. What is the correct answer here? Okay, here the correct answer is option number B here. Okay, so Darwinian variations, they are small and they are directional. Okay, so only those variations which give survival advantage, they are selected in the next generation, they are inherited to the next generation, so they are directional. Okay, nature is only selecting those traits, those genes, those alleles, those variations which give survival advantage. And mutations, they are random, they are directionless. Clear? Next one, Hugo de Vries proposed the idea of saltation based on his work on which plant? Okay, so Hugo de Vries gave the concept of saltation. Okay, so he was working on which plant? So options are pea plant, evening primrose, maize, or Drosophila. What is the correct answer? Okay, option number B is the correct answer here. Okay, so Hugo de Vries gave the concept of saltation. Saltation is single step large mutation which can lead to speciation, as said by Hugo de Vries. The next one, which term did Hugo de Vries use to describe evolution through sudden large mutations? What are the options? Graduation, natural selection, saltation, and genetic drift. What is the correct answer? Okay, option number C is the correct answer here. Okay, so saltation is basically single step large mutation. So Hugo de Vries said this evolution of a species can occur in just single step due to these large mutations. Okay, so he said evolution is not gradual. Next one, according to Darwin, evolution is a gradual process. Okay, means it is slow. Which of the following best describe de Vries' view on the pace of evolution? First one, evolution occurs gradually over millions of years. Means evolution is a very slow process. Second one, evolution occurs through slow accumulation of minor variations. Third one, evolution occurs suddenly in a single step due to large mutations and means saltation. And third one, evolution does not involve genetic changes. What is the correct answer here? Okay, option number C is the correct answer here. Evolution occurs suddenly in a single cell due to large mutations. This was the concept given by Hugo de Vries. Okay, so he said evolution is not slow, it is not gradual. Evolution of a species can occur in just single step due to large mutations, which are random and sudden. Next one, which later field of study helped clarify the mechanism of evolution by integrating ideas from Darwinian variations and mutations? The options are paleontology, population genetics, comparative anatomy, and embryology. What is the correct answer? Okay, the correct answer is population genetics. Okay, so this helped clarify the mechanism of evolution by integrating ideas from Darwinian variations and mutations. Okay, option number is paleontology, which is the study of fossils. Okay, so basically paleontology give you this is the evidence of the evolution. By studying fossils, you can determine whether evolution is occurring or not. Then comparative anatomy, this is also evidence of an evolution. So by comparing the you can say anatomy and morphology of the organisms, you can tell about the evolution, whether evolution is occurring or not. Then embryology, this is also evidence of the evolution. You can study the embryos of different species, whether they have similarities or not. Okay, so option number B is the correct answer here, population genetics. Here you can study the gene frequencies, allele frequencies here. Okay, so with this, we have discussed the questions also. Clear? Now all our questions are correct. So with this, we end this session. I hope the session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.